Okay, back again. I'm recording this on Wednesday morning, the day that the ice storm is moving into the area. All the texts I'm getting are my wife concerned that I'm going to get stuck up here and never be able to come home again. So that's why I stopped a couple of times to reassure her that everything's okay. So let's continue. When conditions break down hydrogen bonds, an enzyme changes its characteristic shape, which is called denaturing, and stops working. So basically, you have a specific pH, temperature, and salt concentrations. If those things get out of control, then the shape of your enzyme can change. And if the shape of your enzyme changes, then it won't function anymore. You have things called regulatory molecules that affect enzymes by binding directly to their active sites or elsewhere on the enzyme. And I suspect, yes, this will be a nice little item to show how regulatory molecules work. You see you have an enzyme. It has to have these two little regulatory molecules attached to it, and then your substrate's attached. Generally what happens in these reactions, if the regulatory enzymes are absent, one of two things will happen. If the enzymes are absent, the substrates may not get there, or the substrates may not be able to attach, so sort of a polarity happens. Whoops, let me back up one. Other times, it might be the presence of those regulatory molecules that stops the substrate from being able to attach. So a regulatory molecule is going to regulate. It's either going to make a process happen or prevent a process from happening. Metal ions or coenzymes that associate with an enzyme are necessary for its functions. Coenzymes, you know these coenzymes. Coenzymes are biologically, nutritionally, excuse me, we would refer to a coenzyme as a vitamin. An example, dietary vitamins and minerals, those are coenzymes that act as controlling factors that help our reactions work. Coenzymes are always organic, so keep that in mind. They can be modified by a reaction, and you have the coenzyme NAD+, and you add electrons, and you get NADH. We will talk much more about NAD+ and NADH in a future chapter when we start talking about cellular respiration. ATP and phosphorylation. ATP functions as a coenzyme in many reactions. Without the production of ATP, you and I cannot live. Used to have a question on my exams that said if you accidentally took a poison that prevented you from making any ATP, what would happen? And the correct answer would be you would die. So, very important molecule. Phosphate group is transferred to or from a nucleotide. Energy is transferred along with it. The phosphate group, I showed it to you the other day. It's a phosphorus ion with 4 oxygens attached to it. Its chemical formula is PO4. Here is the energy in, energy out of the ATP as an energy carrier. You have the energy in, you have the ATP formation, the energy out, and you wind up with ADP. ADP plus phosphate gets here. ADP forms in a reaction that removes the phosphate group from ATP. Metabolic pathways. A metabolic pathway is a set of chemical reactions that looks like this. You have product A. Through a reaction will yield product B, which then through a different reaction yields product C, which through a different reaction yields product D. And in you and I, and in most living things, this cannot be altered. A yields B. If you do not get B, you never get C or D. And this is some of the problems of different things like diabetes, type 1 diabetes, where a person is not making any insulin. It may very well be that they have something going on in one of these early steps, so they don't get out here to where insulin ultimately is formed. And that A yields B, yields C, yields D. Each one of these arrows represents an enzymatically controlled reaction. So if for some reason you don't make enzyme number 1, you never get product B, never get product B, you never move any further. This is a metabolic pathway, a series of enzyme-mediated reactions by which cells build, remodel, or break down organic molecules. Again, there is a linear and cyclical view of metabolic pathways. Again, you start with the reactant, act on it by enzyme 1, you get some intermediate structure. That intermediate is then reacted on by by enzyme number 2 to get another intermediate structure, and then you get enzyme number 3, which produces your final product. And again, here's just the same thing, just written in a circular fashion. There are various controls over enzymes which allow cells to conserve energy and resources by producing only what they require. And there are methods for this, how this is controlled. One of the controls is the concentrations of reactants and products. Concentration is important. Think about it this way. If you drive in to work at 3 o'clock in the morning, how likely are you to have a fender bender? But if you drive in at 8 o'clock in the morning, how likely are you to have a fender bender? At which one of those times would it be more likely that you're going to accidentally bump into somebody else? And I would say 8 o'clock. And the reason is there's a whole lot more cars on the road. If you drive in on the interstate, there's a lot more cars on it at 8 o'clock than there are at 3 o'clock. So the higher concentration, you're more likely to have an accident. So the higher concentration here, the more likely the reaction is to happen. There's also something called feedback inhibition. In feedback inhibition, we actually, the control is actually based on the product. There are types of feedback inhibition where if you've got enough of the product, the excess of the product stops the process from happening. In other types of inhibition, in other types of feedback inhibition, the presence of the product actually starts the reaction. So in example 1, you have a product that when you get enough of it, shuts the reaction down. The other example is the product being present starts it. So in one example, the product stops it, and in the other example, the product starts it. And again, this would be a feedback inhibition, an example. Here's my reaction. We get enough of this product, and it comes back here and stops the reaction from happening. That is feedback inhibition. The electron transfer chain, we will talk more about this again when we talk about cellular respiration. But it allows cells to harvest energy in what is referred to as a manageable increment. There's a lot of energy in a molecule of glucose, but you can't get it all at one time. There's an old saying of the old question is, how do you eat an elephant? One bite at a time. So this isn't the electron transfer chain. There's a whole lot of energy, and we're going to get it a little bit. We're going to collect all of it, but we're going to collect it a little piece at a time because we can't gather it all at once. We're going to get it we're going to get piece by piece. Controlled energy release here, again, we got the glucose molecule. They're burning glucose, and I don't know how many of you knew that, but yes, glucose will actually burn. If you ever see burning glucose, don't touch it, because it will stick to your skin and then continue to burn while it is stuck to your skin. Basically, here's a pile of glucose. What's burning right here will be like a syrupy mess, but you touch it, it will stick to your finger or whatever you touch it with, and it will burn extremely badly. Again, this shows that stairway production. This is the electron transport chain here. And you see how we've got some energy here. We get a little of it here, we get a little of it here, we get a little of it here, we get a little of it here. So we're collecting it. Electrons, hydrogen, and oxygen combine to form water. Water is going to be the last thing to come out of these reactions. Diffusion. Diffusion is a process where molecules will spread through a liquid or gas. If you've ever been at home cooking and maybe you put a cake or a pie in the oven and then you set the timer and you go back to your den or your man cave or your lady cave or wherever you happen to be, watching TV, doing whatever you're doing, working on the computer, and you start to smell the cake or the pie cooking, that's the process of diffusion. Those molecules are being released as they're being cooked, and they are diffusing through the air. They're spreading from where there's a lot of them in the oven to an area where there's not a lot of them. They spread out. Same thing happens when you put a tea bag in water. You see the tea's up here, you see how the color is diffusing away from the bag? That is diffusion, movement of particles from where they are highly concentrated to an area where they are less highly concentrated. The rate of diffusion can be affected by several things. Concentration. If there's a big difference between what's right here and what's out here, they're going to move relatively quickly. As those concentrations become closer and closer together, the movement will slow down. Temperature. Warming things makes diffusion happen faster. So you elevate the temperature slightly and you elevate the process of diffusion. Particle size, again, large things tend to move slower than big things. And when you're diffusing, again, you're having to make your way through a medium of some sort. The smaller you are, the easier it is to find a path. Charge. Positively charged things may move more readily through or across a membrane than negatively charged things. Again, the charge will play a role, and pressure. If you put things under pressure, more things will diffuse. If you were diffusing, again, that tea bag, for instance, there will come a time when in that glass of, or that cup of water, that diffusion will stop. The net movement of things will stop because the pressures are equal. But then if you suddenly take that and you put it in a much larger or deeper vessel, then it will start to happen again because the pressure has been increased. A semi-permeable membrane, we talked about this with the fluid mosaic model. We talked about that it was semi-permeable. Allows some things to pass by and other things not to pass by. This is where we get the concept of tonic or tonicity. What is the relationship of this product on this side of the membrane to this product on this side of the membrane? Three terms, one of which is very easily understood. The other two can get quite confusing if you let them get confusing. If you simply remember that hypo, HYPO, means low, and hyper, HYPR, means high, these things aren't that confusing. Low solute concentrations relative to another fluid is hypotonic. If a cell has 3% salt and an environment has 8% salt, then that cell is hypotonic to the solution. If the solute concentration is the other way, if you've got 8 in the cell and 3 in the environment, then the cell is hypertonic to the solution. An isotonic solution is where the solutions are the same. You got 5% in the cell, 5% outside the cell, boom, isotonic relationship. And this again shows the phospholipid bilayer. It shows things like carbon dioxide, oxygen, different gases, shows water, shows that those things will come through, but large molecules like most ions, glucose, and other polar organic molecules will not pass through. Again, polar things won't, non-polar items will. Again, very big, large, non-polar, very, very large non-polar items won't, but small non-polar things will pass through. Osmosis is the diffusion of water. This is where I get to tell a little story on myself. The diffusion of water across a selected permeable membrane between two fluids that are not isotonic. When I was a student over at UALR, I was taking an organic chemistry class. And I was in the class. There were 30 or 40 other people in the class, and I was the oldest person in the class. I came to school late. I didn't start college until I was in my 30s. So my professor was up there, and he said something about diffusion, and he said, and we all know what diffusion is, correct? And I shot my hand up, and he said, yes, Mr. And he looked down the seating chart, and he said, yes, Mr. Russell, what is diffusion? And I said, Professor Yang, the diffusion is the movement of water across the selectively permeable membrane between two fluids that are not isotonic. And Dr. Yang stood there with his hands crossed, looked down at the floor, and just shook his head and said, another biology major. And he sat there for a second, and he looked up and he said, now class, why do I know that Mr. Russell is a biology major? Dr. Yang knew I was a biology major because I had been working in the biology department for 2.5 years, and we had been to departmental and school meetings together. He knew I was a biology major. But the point he was making was this. This is a biology definition of osmosis, the diffusion of water. The chemistry definition is diffusion of a solvent. Now, for biology purposes, universal solvent is biology. So this is a fine definition for biology. But if any of you ever happen to be in a chemistry class and the guy and the instructor says, that was kind of sexist, I apologize, the man or woman person, the instructor, asks what diffusion is, you say diffusion of a solvent across the selectively permeable membrane. And they will go, oh, great, that is a wonderful definition. Where did you get that? And you go, yeah, Mr. Russell told me that in biology class. If the extracellular fluid is not isotonic, cell volumes change. In a hypertonic fluid, cells will shrink. In a hypotonic fluid, cells will swell. So you'll probably see on your Chapter 4 exam, on exam number 2, there will probably be a question at some point that says, if you place a cell in a hypertonic solution, what would you expect the cell to do? And you could say shrink, you could say swell, nothing, turn blue. You may ask a bunch of different things, but the answer would be, if you place a cell in a hypertonic solution, it will shrink. Again, this is a wonderful little experiment that you actually can buy these apparatuses. You can buy this apparatus to do this. It's a really neat experiment. You have a semi-permeable membrane on one side in the middle of the U. You make up 2 solutions that you know one has a certain amount of fluid, the other will not. And you simply put the 2 fluids equal amounts. You put the lower concentration here, the higher concentration here, and then you come back about 30 minutes later, and you will see the volume has changed as the product, the water. See, what's happening here is if you count these little dots, the little dots aren't moving. Only the water will move. And the water moves kind of slowly, so you can measure out your volume. You can pour 50 milliliters over here, and yeah, the water will start leaking a little bit. And then you pour your 50 milliliters over there, you wind up at the same level here. And then over time, the water's moving. And if you take the time to count these little dots, I'm not going to do that. But if you take the time to count the little dots on this side of the membrane and this side of the membrane, you will see that the number of dots is equivalent. Now there's still 3, 6, 9 dots, 3, 6, 9 dots. But what you notice about these is you see how closely packed together they are and how they have started to disperse. Now, again, depending on the pressure that you have and some other factors, this actually may continue till you start losing volume and it starts to spill over the top. This is an example of what happens in red blood cells. This is a normal red blood cells. These are in isotonic solution. That's what a red blood cell is supposed to look like. You see it's a little disc that's been pinched in the middle. This is what happens in a hypertonic solution. Remember, in hypertonics, things are going to shrink. You see how these nice little round, sort of almost donut-looking shape things. This one's really flat. This one's got a funky shape. These are shriveled and just nothing. But then what happens when you put it in a hypotonic solution? Look, they're starting to swell. They no longer have the indentation. And look, a couple of them have actually even burst. So that is the effects of tonicity on cells. In plant cells, there's something called turgor pressure. That's the pressure that the fluid exerts against the cell wall or a membrane or other structures. Osmosis continues until the two fluids become isotonic, unless the pressure against the hypertonic fluid counters the movement. That's what happens. If we back up one, animal cells will do this. If you place them in a hypotonic solution, they will swell and burst. Plant cells, on the other hand, will not do that. Because of the presence of the cell wall, the plant cells are going to spread to a certain level, and then they're going to stop because the pressure is preventing any more osmosis from occurring. Again, the effects of tonicity. This is what happens when you put a cell in this cell. If you'll notice, it's full. All the little chloroplasts are spread way out. This is an experiment we used to do with Elodea leaves. Elodea is getting harder and harder to find, but it's the little one of the plants that people put in ponds or fish ponds and that kind of thing. But it's getting harder and harder to find locally. But this one they put on a slide of Elodea. That's what it's supposed to look like. You see how the cell is full and you got these little chloroplasts all over the place. Here, they probably put salt water on it, I suspect. And the salt water has drawn all the moisture out. And you see now the membrane, you can't even see the membrane here because it's pressed firmly against the cell wall. Here you can see the membrane, and it's shrunk considerably, and all of those chloroplasts are now compact and can't move any. That is the effects of tonicity. The water has been removed. Those cells, the cell wall hasn't changed, but inside the cell has shrunk. Gases, water, and small nonpolar molecules can diffuse. Most other molecules and ions cross only with the help of transport proteins. And each type of transport protein moves a specific ion or molecule across the membrane. So think about it. Think about how many different molecules have to get into and out of a cell. If each one has their own transport protein, you can now begin to understand how many transport proteins there must be in the surface of a cell. Passive transport is where things are controlled by a concentration gradient. You got a bunch here, got a little bit here. Things naturally are going to move down that concentration gradient. Passive processes just happen. They don't require any energy transfer. But, and again, glucose transporters. On the other hand, we also have processes called active transport. Active transport is where you're not going down the concentration gradient, you're moving things up the concentration gradient. While things will roll downhill spontaneously, you've got to put energy in to move things uphill. So passive transport, no energy input. Active transport, concentration, you're moving against the concentration, so you must have energy input. Here is just simply an example of passive transport of glucose. You see there is this is your transport protein, and you see the glucose molecule fits inside there and then is pushed into the cell. That just happens through, again, you've got a lot of glucose out here, no glucose, so you've got the passive transport system. It's just going to diffuse through. Active transport, on the other hand, you've got this calcium inside the cell. You want to get the calcium outside the cell. So there's calcium. If it was just a passive transport, the calcium would just move through. However, that's not what happens. The calcium must be pumped out. And here's that energy. ATP fires the reaction, reduces it to ADP. Endocytosis is movement of things into the cell. And once we start talking about endocytosis and exocytosis, we're now talking about things that are too large to simply pass through the membrane. They have to be taken in and taken out. Endocytosis is a process by which the cell takes in a small amount of cellular fluid and collects those things that it needs. Exocytosis is the use of a vesicle. Remember, we talked about a vesicle the other day. We were going to expel the vesicle's contents to the extracellular fluid by merging the vesicle with the plasma membrane. And I'm sure this is going to be, or this next one will be a picture of how that works. Phagocytosis, also sometimes pronounced phagocytosis. I learned phago. Maybe phagio is a more proper pronunciation, but remember, I went to school in the South, so there you go. But phagocytosis or phagocytosis is an endocytotic pathway by which cells, such as macrophages and other white blood cells, engulf particles such as microbes or cellular debris. So here is number one. We have exocytosis. There's a vesicle. Here's my vesicle. Contains a bunch of stuff. I don't know what's in there, but it doesn't really matter. At point number two, the vesicle's membrane fuses with the plasma membrane. And as that fusing occurs, as the membranes fuse, the vesicle contents are released into the extracellular fluid. So, last couple of things. Testosterone supplements have been used by athletes for some time. Why do athletes use them? Athletes use them to get better results from their exercising. They use testosterone instead of estrogen. Testosterone is a muscle building hormone. Estrogen is a fat building hormone. So if you're trying to, lift weights, give yourself, if you're a dude and you want to have a really, really big chest and really, really big arms, you don't want to use estrogen, because if you use estrogen, you probably wind up with a really, really big belly and a really, really big butt. You don't want that if you're into bodybuilding. Should the cause of liver failure be used as a factor to prioritize liver transplant wait list? Why or why not? This is simply a philosophical question. If someone has liver failure and the liver failure is self-induced or self-perpetuating, if they're an alcoholic, a bad alcoholic who's been an alcoholic for years, should they get priority over someone who has liver failure because of a genetic defect or the fact that they got hepatitis at some point during their life? Of course, you also have to consider, why did they get the hepatitis? So again, that's something to think about. When you hear about somebody being on the wait list, again, there was a big controversy. Oh golly, it's been 15 or 20 years ago. There were a couple of very prominent people. One was Mickey Mantle, a former baseball player, Hall of Fame baseball player, and the other one was the actor Larry Hagman. Both at about the same time got liver transplants. And there was a lot of controversy in America at the time as to why did these two very prominent Americans, how was it that they jumped to the front of the liver transplant list ahead of a bunch of other Americans when they had both been alcoholics for 40 years and they had basically destroyed their own livers? Why did they get livers before other people did? And the official story at the time was, well, the livers that became available, those two gentlemen were matched for, and those livers didn't match anybody else. I don't know. Could that be the truth? Yes. Could it be that because they had money, they were able to get through the livers before other people? I don't know. I wasn't on the board that decided who got them. But that was a big story at the time, is we've got thousands of people on the liver transplant list, and these two celebrities basically get livers. And it really became bad when one of them, within about 6 or 8 months of getting the liver, I think, it may have been a little longer than that, basically died. And the reason he died is because he never stopped drinking. If you have any questions, please contact me via either email or the course content link, not the course content link, the course messenger within the contact area of the blackboard shell. Again, apologize for the fact that this is three very sort of disjointed recordings, but again, wife is at home with the baby and really concerned about the weather, so she's wanting to make sure that I'm aware that the weather's moving in, and hopefully I won't get trapped here. So have a good day. See you next time.